BIanka. I'm wearing a light blue and white striped summer dress with no sleeves, and my hair is red-brown and in a plain ponytail. And I'm sitting to the viewer's left of Matthew.
Yeah.
And you would just say something.
So like I'm Matthew. I'm wearing white shorts with brown shoes and a brown and white striped short sleeve shirt. I have brown hair that is spiky, and I'm sitting to the
Viewers,
the
right. Yeah. Thanks. So we were just debating the positioning of orbs in the story of the visit to the gallery, and the orbs being building blocks of sugar. So in my mind, they could go somewhat early in that story, because we start the journey with sucrose, sugar as we know it, which could be, fairy floss and everything that's fun and energetic and wonderful about sugar, and looking at that as then a building block for sugars as they're processed in the body. So I'm thinking about these orbs. These big white structures came from a process of folding circles, circles being representative of, the most I could reduce a shape to represent a sugar molecule or a cell or the circle of life, like a circle being very representative. And then just folding paper circles over and over and over again, trying to make different combinations, and so that's how these were kind of conceived. Trying to make building blocks that look the same but they're not. There's a difference, a variation to them. So I see in the journey going from sucrose to understanding building blocks, and then maybe the densities of cell tissue, which you've taught me about. And so the layout of those is in relation to the tissue types and how spread out and how dense they are, and having a disease centre that's really quite dense. It has the deteriorated or variations in there as well. You're looking at it from a different point of view of looking at the pillars and those kind of things first. Tell me about tell me why you're looking at orbs being later on in the story.
For me, the orbs represent, I guess, the different sugars or the complex sugars that I look at. So as you've already said, simple sugars like sucrose or glucose are being metabolized by our body or being processed, and they get built up into different simple sugars that get like building blocks like Lego in our body, and these different building blocks can then change depending on I guess what our cells are doing at the time, if they're growing or if they're separating out into new cells, or if they're doing something like, for example, in cancer, they change as well. So looking at that from the aspect, the protein pillars have sugars on them,
Mm-hmm.
these complex sugars on them, and that aspect really interested me in terms of having that first in the story, because understanding the proteins first before we get onto the sugars is something important.
Yeah, so throwback to high school biology classes, you learn that a cell has a cell wall, and out of that cell wall these proteins can protrude. So I remember drawing them, in year 12 biology, getting ready for exams, drawing them as these sort of oval or dome-shaped protrusions from a cell. And what I've learned from you is that the sugars attach at those proteins, and while I've represented them here as one really sturdy protein base, it's powder-coated steel base that's really strong and sturdy, that's attached to the floor of the gallery, like as if the gallery floor is the cell wall. And what I have is an idea of a sugar per pillar, when actually I think you've told me you can have, say, 50 sugars coming out of or attaching to a protein.
Yeah, or 3,000.
3,000, that's incredible. And the reason for keeping this kind of straight line going on is because of the graph that you showed me We did a Zoom and you actually opened it up and you were showing me how you can look in at the different kind of levels of that, and thinking about it like a line, sort of bar graph, but a line graph. You're looking at very much black lines of varying heights on a white background. So that was the initial premise for the shape and the height, along with the overall theme of forest, because of the description of sugars coating a cell perhaps being like a forest coating, and things that can move and there's communication happening. So this idea of being able to traverse a space like a forest kind of space, so these being say tree trunks is...
And also the complex sugars I look at, they can branch. So like
a tree can branch,
like a
and
branch,
branch
yeah.
and branch and branch.
And you know, the change between normal and disease can be, in terms of its branching, can be the same complex sugar in terms of what it's made of. But the branch can be different orientation, and that can then change everything.
This is really fascinating. This is something that I really got stuck on, that these structures, going back to chemistry lessons, think of chemical compounds, how we might draw them. You look at describing through a series of squares and circles and triangles, and you look at describing that structure in a way that's very similar or familiar to a chemical compound. And you have this branching that happens, and you have extremely complicated long chains of sugars. And then you've got one tiny branch change, which is the difference between disease and non-disease. And I found that so fascinating, that change was so minute and had such a severe consequence in some cases. And that it's such a tiny thing for you to be searching for, like the difficulty of your process in having to look for that is really fascinating to me, and why, with the pillars, for example, you've got this view of them that is completely, say, normal. You can't tell that there's anything, and then you'll take another look at them from another perspective and you can see this variation, and sometimes it's quite an obvious variation, and for me it's indicating, say, disease state. Some of the other variations are more subtle, and some of them it's really difficult to find, because your process, it's a difficult process to try and find.
Which is often the case in biology, in terms of certain diseases are easier to detect and prevent, and therefore, and then some of them are much harder, like ovarian cancer, which is what I look at. So
so you do consider things that aren't just ovarian cancer. You do look at some things like osteoarthritis in the knee and some other things, but you talked to me about blood sugars, blood types, which is really an interesting way to kind of sum up what it means to have sugars in your body.
Yeah.
Can you tell us about blood types?
Yeah.
Everyone has a blood type.
Sure. So these blood type sugars, they're different to the sugars in these diseases that I'm looking at. So they're made differently, but it's still the same concept and still the same building blocks. But the 4 blood types that we have in the world, each of the different blood types have a different sugar pattern, and that's how transfusion was discovered, in terms of how we can then transfuse between the same blood type between patients, because they have to have the same particular sugar blood type. And AB, for example, has bits of A and bits of B combined, so they have both sugar blood types.
Yeah, it's the idea that sticks with me about the blood type in sugars, is our identity is expressed, the identity in terms of our blood type, something that's quite critical to our health and well-being, is expressed purely by the fact that there's these sugar coatings on cells and that they're different.
Yeah. But even more, like recently with COVID, obviously, and viruses and HIV, for example, we have these viruses have these proteins on their cell walls, and they have sugars on them, and the sugars is how they enter our cells and how they infect us, because they bind to our cells through the sugar pattern and enter us. So that's how viruses work, basically.
You're one of a handful of people in the world that study the type of sugars that you study, or is it sugars and glycans?
So it's the the technique that we currently use, we're a handful, about 4 or 5 of us around the world that use this technique to study these sugars in terms of using samples from surgery, and the way we can then spatially map these sugars on tissue sections.
It's incredible that that's happening right here in South Australia.
Yeah.
Do you want to tell us anything else about the research side of things that you do?
Yeah, so we get a lot of samples from surgery, as I said. So particularly the cancer samples and people that have knee replacements, so we for the osteoarthritis samples. And we basically, we section these, or we slice these tissue sections up and put them onto slides. And we can then spatially map, as I said, these sugars using a technique where we use a laser. And we can then basically get these peaks or these big pillars and identify where they're located in the regions, and use clinical information then to determine what the cause is or what's happening. So, for example, I compare early and late stage patients to try and then determine, if there's a difference between them, and then also then to see if we can then see if we find a sugar that's in early stage and that isn't in the late stage, and we can then try and detect in the clinic ovarian cancer before it spreads.
Yeah, so this could be applied in the future too, or perhaps it's being applied now, but detecting the sugars so you can make an identification early.
Yeah, so it's like a signature almost.
Yeah. The process of the identification, like I said, is tricky and hard, and I mean, dealing with something that's so small you can't see it in any format. You can't put it under a microscope and look at the sugar structure. And you've got this system of using a laser to identify, and so it was that idea of using light and lasers is why there's elements of light peppered through this work as well.
In terms of scanning and trying to detect something that's very hard to detect as well. Yeah.
I wonder if, because you and I had a cup of tea and started a conversation and decided to hit record, I wonder if there's anything maybe more background and broad we need to consider about sugar. We use the word glycan, and I don't know if we've really discussed, described what a glycan is.
Yeah. So the glycan is a scientific term for a complex sugar. There can be multiple different types of glycans. The ones we're talking about today are on proteins, but my particular interests are end-linked glycans. So these are glycans that are attached to, or sugars attached to asparagine residues. So there's 20 amino acids in the human body, and asparagine is one of those amino acids. So on proteins. And then there's also O-linked glycans, which can be attached to serine or threonine residues. So there's another two amino acids it can be attached to. And it's just processed in a different way in terms of our body and our genome. So there's about 700 different glycan genes that are currently working in our body that are expressing all these enzymes that are processing the sugars and attaching things. So
I did read your research paper that you gave me on your N and O-linked glycans. I can't say I understood everything, but there are images in there that are sort of being shaped, and they have different areas that are showing through colour, showing sort of the lighting up of certain areas of the cell section. So I was really focused on the imagery in that.
And then there's also things that are in our food products like dextrin, which is a long chain of glucose subunits which is put into our food, which we also use for our research as well.
So we've gone on this journey then from sort of sugar as we know it, sweet sugar, sucrose, being digested, being converted, being a building block. And this process of identification and searching and the consequences of that are all things that we've kind of explored through this process.
So in terms of health benefits, I think, for like sugar. We could discuss that as well in terms of, and really question, like, are we overloading ourselves with too much sugar, and is that causing our machinery and processing machinery to fault, and is that causing us to have disease in certain areas of our body, or is it, or is it the sugar we consume ourselves? Is it something else that's happening in our body that's causing that fault, and therefore when we consume too much sugar, it builds these incorrect mistakes.
Yeah, like a processing line that's having to run so much faster and work so much harder than it really wants to be working.
Yeah.
It just creates faults.
Yeah.
Statistically will create more
and there will be
faults.
errors.
Yeah. And like we've discussed, such a minute change on something that's so complicated can be the difference between a regular sugar and perhaps a diseased sugar.
Yeah. So, for example, diabetes is, the fault is within the machinery, not the actual sugar you digest. So,
as opposed to processing it incorrectly, as part of a normal process through the body and processing it and putting a sugar out that's that has an error or inconsistency to it. Tell me about how many glycans exist.
There's currently several different libraries, we call them. So different databases which currently have them on there, but it's quite varied in terms of the literature and how many there are, because there's no consensus between the libraries.
Yeah.
But there's well over 300 at least, in the cancers that we know of.
Yeah.
Well, because of the different types of branching, for example, as well as what they're made up of.
Mm-hmm.
So there's quite a lot. And our technology is just getting better and better in the way we can actually detect more and more since the past 10 years, because this particular field of research is quite young as well in terms of these two particular sugars that I look at.
Yeah.
Even though sugar research has been around for quite a while, it's still quite young, in its infancy. So at least, especially in Australia as well, because there's not many groups that do it in Australia. So it's mostly European groups and American groups that study it.
It's an exciting time to be in that field when you're sort of looking at methodologies and processes, and I imagine there's some room for creativity in terms of shaping how things are going. And I know you're quite active in the world of writing research papers and sharing your knowledge. I was thinking about this in the wee hours of this morning. I did have an alarm set in the hour of 5 this morning. And I was thinking about my first studies after leaving school. I'd done some work experience in that biomedical field, and that's what I went to study, and worked with preparing histology samples.
Yeah.
And they're the types of samples that would arrive in your laboratory for testing. And I was thinking about that process, and it's why some of those histology images are so often in the way cell structures are, in a sliced sample. And that kind of sampling is kind of where that image of the recording of the shadows created by leaves moving.
Yeah.
To me, they look a lot like histology samples that I remember. But I was thinking about, I switched out of the studies in biomedical science and I switched into food science. And biomedical science feels like a very regimented, process-driven field. And it's in food science, if I was to be asked to sort of look at the shelf life of a product, increase the shelf life of something, I'm treating it very much as a scientific experimentation, and that it's got quite a lot of rigour to the process, and I'm looking very much at the numbers. I'm changing the gases in a packet, I'm recording the microbial samples, and it's a numbers game to get this sweet spot. But at the same time, I might have been asked, it's not a field I work in anymore, to look at new uses for a product, perhaps even like a waste ingredient. And then that's very much takes it into this world of caring for the ingredient and wanting to represent that, and then marrying it in. For the first time, you start thinking about people and what is their perception of it now, and what do they like about it, and how do you make this marry. And I was thinking about the creativity that comes from that process of really just research-based creativity and having to think of new things, and how that process has a lot of movement for change, and how that's probably very similar to a lot of my making process as well. It's got quite a lot of rigour and research, but actually within that there's still so much room to keep exploring. And then I was thinking about how that ties back to the fact that your field is so young, and there must still be that room for creativity in how you're going about things, but yet it's still so much rigour around the processes.
Yeah, like most recently, a PhD student I'm supervising, for her project she's using osteoarthritis samples, and last year the tissue kept falling off the slide during the heating step because it's bone, and the bone doesn't stick that well to the slide. So we had to then creatively think of a solution for that, and we ended up publishing last year the solution to that, which was coating the slide in gelatin.
Yeah, because normally you put a tissue sample in paraffin wax or some sort of wax that normally would adhere. Yeah,
so you paraffin embed the tissue sample. Before that you form and fix it, so you preserve all the proteins, and they cross-link. And during the experiment you uncross-link those proteins. But to section onto the tissue, the paraffin isn't that sticky really, if you think about it.
Yeah, and it's more there to hold the cell and the structure that
the structure
it
in
holds, so that
place.
you can take this micro thin slice and then look at it in that same, you haven't crumpled it all up through that process.
Yeah.
Yeah, they stay there. They stay there just because they stay there.
Yeah.
And I can understand what you're saying about it wanting to move. So that process, is something that hadn't been done before. So you've really had to invent something.
And gelatin was the solution to that. So the gelatin's very sticky. And there's other, possibilities of egg white is quite sticky as well. And you could coat the slides in that and try and get the tissue to stick better. And it ended up working. So.
Yeah, and so now you've published something on that.
Yeah.
Fantastic.
So it is very, in a way, it's very an artistic process in terms of the way you've got to think.
Problem solving. I think problem solving is a key thing for both of us.
Yeah, exactly.
Yeah. We've had lots of conversations. We've had lots of conversations. We've had lots of conversations that go on for quite some time, to the point that we'll have a Zoom, and then we'll kind of wrap it up, and an hour later, like, now we have to really stop.
Yeah, exactly.
Yeah, I think the research you're doing is fascinating, and I think it's more so because there's a familiarity to this world of science. Where I can't understand it to the level that you're working at, but there's a familiarity to processes.
Yeah.
And certainly previous studies have helped me in that field. It's wonderful to chat about this. Is there anything else you want to?
I think we covered most things.
Okay.
Maybe not in the right order, but.
I know. I think we started because we were already chatting and we pressed record, but.
I'm sure you can edit.
Yeah, or maybe I won't. We'll just let it roll.
You just keep